This experiment is an introduction to enzyme-linked immunosorbent assay, also known as ELISA. This method is used to detect the presence of a certain substance in a liquid sample. In this module, we will be simulating the detection of an STD. First, label an Eppendorf tube from the rack, as well as your half of the 8-well strip with your initials. Using a micropipette, transfer 50 microliters from the tube to your first well. Pair up with another student and record their name. Transfer the contents of your Eppendorf tube into your partner's tube and mix. This mixing of fluids acts as a model for what happens when bodily fluids and STDs are exchanged between partners. Transfer half of that mixture, about 750 microliters, back to the empty original tube. Transfer 50 microliters of this mixed sample into your second well. Pair up with another student and repeat, transferring 50 microliters to the third well after your second partner, and 50 microliters to the fourth well after your third partner. Share the 8-well strip with your lab partner. Each of you will only use 4 wells. Incubate your protein antigens at room temperature for 5 minutes to allow the protein samples to bind to the well. To wash, first tip the strip over onto paper towels and gently tap the strip to remove the liquid. Make sure that no liquid splashes back up into the wells. Using a new transfer pipette, add wash buffer to each of these wells. Tip the strip over again onto clean paper towels and tap gently. Throw away the wet paper towels. Repeat the wash one more time. After the second wash, add 50 microliters of the primary antibody to each of the 8 empty wells. This antibody binds to the protein antigens. Incubate at room temperature for 5 minutes to allow the antibodies to bind. Wash away the unbound antibodies by performing the wash 2 times. After washing, add 50 microliters of the secondary antibody into each of the empty wells. Incubate at room temperature for 5 minutes to allow the secondary antibodies to bind to the primary antibodies. Wash away the unbound antibodies by performing the wash 3 times with PBS without tween. Add 50 microliters of the enzyme substrate to each of the wells. Incubate at room temperature for 5 minutes to allow the enzyme to react with the substrate. If the contents of the well are blue, this indicates a positive result. Clear contents indicate a negative result. Here is a quick animation to help you understand how ELISA works.